This is a production of Cornell University.
Hello, everyone. Thanks for coming. My name is Phillip Lanza. I'm a PhD candidate with Louis in the Farmers Data Lab. We work with farmers to kind of help them experiment a little better, get more value from their experiments, more or less. So I'm going to talk about first some of the projects I'm working on, and at the end, go over some more of the stuff our lab does. What I'm working on is applying observational research techniques to agronomic systems. They work well in medical trials where things are complex, the data is kind of messy. There are data-driven approaches, and the big thing that we need to do this is context. So, in medical, it'd be like, does this person smoke? What's their age range? All these things. In farming, we use relevant biophysical and environmental data that let us kind of objectively say, okay, well, this happened. What context did it happen in? So the challenge to that in the projects I'm doing, one is a way to help get this data to where we can use it. The next is showing something we can do with these data. And the last is doing some analysis to show that it can kind of help, hopefully, improve kind of experimental interpretation and that sort of thing. So the first thing I did was design a system to help us get all these contextual data. So any kind of agronomically relevant environmental data like weather, terrain data, soil data, all these things. This lets you do it in an easy to use interface. It's web-based, you pull it down and it generates everything in a consistent format. Next thing, building an in-season anomaly detection system to identify areas at subfield scale, right now 3-meter resolution, of anomalous crop response. So given all these covariates, it should be responding this way based upon NDVI. This isn't, and then identify that. So then we can identify these areas and send scouts out there to find them, or find out, this experiment had a bunch of bad areas, so maybe it may not be the best. Last thing, I'm doing a comparison using the anomaly data on a commonly accepted correlation of the great, soil health versus yield. I'm using production environment soil health data from Joseph and Harold's group, which thanks for letting me use. And then I'm going to analyze it where areas have no anomalies versus like varying degrees in C. Like my assumption is that no anomalous areas will be stronger correlation. Then I can show like, when we account for these things, we can get kind of better experimental design and plan things better and just account for like kind of uncertainty. So lastly, we're a Farmers Data Lab. This is the website. If you like QR codes, here's the QR code. We're transdisciplinary. We co-create solutions with farmers using agronomy, data science, GIS, social science. Here's some cool lab members. We have a new postdoc, Abdel. We're growing. We're getting some new members in the fall. I'm looking forward to working with. And if you have any questions about what I'm doing or what we're doing, I'll take them now. Yep.
Phillip, how many farmers are you working with?
What's it, like 12 right now? Is that right?
We have 24 sites, but 12 farms for farmers.
Yep. So we're trying to grow. If you have any farmers, who might want to work with us, let us know.
Do you anticipate increasing that in the future? Is that like something that's expanding, or is it 12 a good number like that?
From my point of view, the more the better. A lot of the techniques I use are data driven, so the more the better. And then we'll get, right now our forums cover a lot of the state, but the more representation you have, kind of the better data you get. And then the more people we work with, the more people we can help, hopefully. So. Sure.
What types of cropping systems are you working? Like what are the farmers growing, and roughly how large are the farms that you're working?
Yeah, sure. There are a lot of dairy farms. They're growing, grain corn, stover corn. The anomaly detection stuff, I'm going to be working with soybeans, but pretty much any grain crop. That's what I'm trying to work with until we can work with other higher value crops, maybe in a couple of years or so. So, yeah, field crops, agronomy.
Thank you very much. You said that you're using this information so you can design better experiments. But that, to me, doesn't seem like the kicker. You kind of want to get that out to the farmer. So how are you actually making that linkage and taking this information to inform the farming community itself?
Sure. Well, one example, what we work with is a little different. We don't impose experimental design. We work with endogenous experimentation. So the farmer, some do like side-by-side strips, which is great. Other ones we've worked with in the past have designed their, say, control around the border of a field, which, as we know, isn't like standard growing conditions. But what I'm saying with the context, we can pull down this to give us objective data that we can then use to kind of explain a little more about what's going on. And then the anomaly, we can go in the past and say, okay, well, this experiment didn't turn out exactly how we thought. Looking at these anomalous areas, maybe their treatment area had a bunch of them within it. So it wouldn't be kind of standard crop response, which we can use to say, okay, well, we better interpret this. Going forward, if we consistently identify anomalous areas, it's probably a biophysical cause. If we do help them design their experiment, say, hey, don't put your thing here, put it here. So did that kind of answer?
That helps a lot, and you're working right with them in the first instance. Yeah.
Sure. All right. Thanks, everyone.
So my name is Becky Stup. I'm a first-year master's student studying in Tony DiTomaso's lab, and I'm here to talk about my thesis project, which is optimizing field margin management for weed control and ecosystem services. So why would a farmer want to plant something in the margin of their field? So biodiversity plantings are very well studied at this point. They improve biodiversity across multiple trophic levels, as well as provide various ecosystem services such as nutrient cycling, water filtration, and more famously, pollination and biocontrol of crop pests by providing habitat for beneficial arthropods. And while the seeds can be prohibitively expensive at times, historically, funding for these plantings has been available to growers through programs like the NRCS Conservation Stewardship Program. I am not sure about the current status of that, but we know from talking to the NRCS that New York farmers tend to put these plantings way out in a corner of their field rather than close to their crop monoculture. So what are some barriers to farmer adoption that we're facing? Right off the bat, there's a lack of research about how field margin plantings affect weed communities within the crop. Especially in New York State, a lot of this research happens over in Europe. So we're not really sure what this might have or what might happen with this. And additionally, farmers have raised concerns to members of our research group, such as, are these plantings going to act as reservoirs for weeds? And is there a chance that these beneficial plants might move into the crop and then act as weeds? So in order to address these concerns, I am comparing weed and arthropod communities in both planted and conventional margins. So I have 2 cornfields, one up in Musgrave, one down the road in Varna, and I've established wildflower plantings and grass plantings, which are either mowed according to NRCS guidelines for weed suppression when they reach a certain height, or they're left unmanaged for the rest of the season. I also have unplanted conventional margins, which are mowed when they reach a certain height. Some are mowed every 3 weeks, regardless of height, and some are sprayed at the same time as the corn, so at about V3. And this is done to just mimic what farmers are actually doing at this point in time. So one thing that I want to get into before I start talking about my actual results from the first year is that these are often perennial plantings, and they don't do much during the first year. So over here on the left, we have an example of great establishment. Most of those plants are wildflowers, but you'll notice a lack of actual flowers. And we know from previous research in this lab that arthropods tend to show up more when there are actual flowers rather than the species itself. All that to say, arthropod abundance did not differ across treatments in this year. Also, if you're going to have a weediness problem, it's typically going to be in the first year because the native perennials do tend to be outcompeted by aggressive annual weeds. So this is the density during the first year of establishment, so the number of weeds in half-meter squared quadrats. And as you can see in July, the main difference that we're seeing is that the glyphosate has significantly less weeds than any of these planted treatments on the right. But by the time you get to September, there's basically no difference across treatments. And from an agronomic perspective, I would argue that September is the more important month to be looking at because that's when a lot of our problematic weeds will be going to seed. We also collected biomass data. So the red is the broadleaf weeds, green is grass weeds. On this side are the conventional unplanted margins, and this side is the planted margins. And you'll notice that each half has 2 tall bars and 2 short bars. I know this is a complicated graph for this presentation, but the main difference here is the mowing, which is pretty intuitive. There's less biomass when you mow, but we're also not really seeing a difference in the unplanted versus planted strips. So if you're a farmer and you're concerned about weeds in your margin plantings, at least in the first year, it's looking like those fears are thankfully fairly unfounded, as long as you're following NRCS mowing guidelines. I'm also continuing a project from a previous graduate student in our lab, Sophie Westbrook. She was looking at how the seeding rates impact establishment, and I just wanted to show you what these plantings look like about 3 years in once they're actually established. We are also starting to see very ecologically important species like butterfly milkweed start to show up, which we haven't seen in previous years, and we're supporting fairly healthy arthropod communities. So thank you.
Wondering, how does the implementation works? Is it broadcasted?
Yes, it is.
Actually, dead machinery there, or?
So yeah, we have been broadcast seeding. I know some companies do drill seed, but especially with the grass seeds, there tends to be issues with them going through the hopper because they're kind of fluffy.
Okay.
So yeah.
Really great talk, Becky. I'm wondering, has there been any work mixing annual and perennial wildflowers within a mixture so that you can see the benefit of the first year?
Not in our region that I'm aware of.
Becky, you said like not a lot of benefits from the first year. I was thinking like that's covering the soil, right? That's like land cover. So that's like we talk about like protecting our soil.
Mhm.
Yeah, that's a really nice benefit to have.
Yeah.
You know, it's not doing what the NRCS is maybe the main purpose.
Yeah, that's a good point.
Hello, everyone. My name is Preetaman Bajwa, and I'm a first-year PhD student with Dr. Vipin Kumar. And today, I'll be presenting you some of the studies that we conducted last year as the integrated weed management approach in corn and soybean. So corn and soybean are one of the major crops, and weeds are one of the most significant pests causing the yield reductions. But what farmers are doing these days, they are heavily reliant on the chemical weed control. So last year, 96% of the corn fields and 98% of the soybean fields received the herbicide applications, which is huge. And with this amount of over-reliance, there is a problem of herbicide resistance. So there are currently 533 unique cases of herbicide-resistant weeds globally to 168 different herbicides. And also, since last 3 decades, we have no new mode of action for the herbicides. And over the top, we have new problems. The palmer amaranth and water hemp, which are the most problematic weeds in the Midwest. They have been recently identified in the northeastern states, including New York. And New York populations have reported the glyphosate resistance and also potential multiple resistance to HPPD and photosystem 2 inhibitors. So what is the need? Today we are heavily reliant on chemical weed control, but it is a time that we switch to a more balanced approach for the long-term environmental and economical sustainability. So keeping that in mind, we designed some of the studies. The first study, we are looking at the cover crop termination timings along with the herbicide strategies for the weed suppression in corn. So we had 2 termination timings, early termination and planting green. Early termination, we terminated the cover crop 2 weeks prior to the corn plantation. And in the planting green, we planted corn in the standing cover crop, and we terminated three days after the corn plantation. So it is said that more the amount of biomass is related to heavy, more weed suppression. So at the time of termination, we have approximately 2.5 times higher cover crop biomass in the planting green, which we also observed in the weed suppression. So this is the first is the early termination with Roundup, no pre or post herbicides. And this is the planting green with effective pre and post herbicide. And in the another objective, we are looking at the 3-way approach with the culture practices with the cover crops, then in season targeting the weeds with the pre and post herbicides, and in later season, we are targeting weeds with the seed impact mill. So we know the cover crops and herbicides, a little bit about the seed impact mill. So seed impact mill, it is the attachment behind the combine that it has these mills which rotate at a very high speed. So they pulverize the weed seeds coming along with the chaff. So main objective is the weed seed bank management so that the weeds that are harvested, they don't go to the weed seed bank and become problematic for the coming seasons. And for the third objective, we are comparing different non-chemical and chemical treatments for the weed seed bank management. We are using the interromower and electric weeder with the mat, and seed impact mill and herbicides. So seed impact mill, I have already discussed a little bit about the interromower. My videos are not working. So interromower, it has the blades here, rotating blades, which rotate at approximately 1800 RPM, targeting the weeds in between the crop rows. So this is how the treatments look like after the application. So this needs the video. No. So this is the electric weeder, the weed zapper that we are using. This is the generator behind the tractor, and it gets the energy from the PTO shaft of the tractor, and the copper rod in front of a tractor. It gets electrified, which targets the weeds which are above the soybean canopy. So this is how the damage done to the weeds. This is the velvet leaf, and this is the stem of the lamb's quarter, completely smoked out. So a bunch of herbicide studies also we are conducting. So here we are evaluating the crop safety and the weed control with the different doses of the metribuzin. So metribuzin is a pre-emergent herbicide. So we plant the soybean, we spray the herbicide, and before the crop emergence. So 280 grams per hectare and 420 grams, this is the normal field rate the farmer use, but they complain that there are some weed escapes with that. So we are evaluating the different doses where we can optimize the metribuzin rates, and we also not harm the crop. So we observe that when we go higher, we have a good weed control, but when we go much higher than the label rate, we have the lower stand count and also the reduced growth. So these are the pictures 5 weeks after the spray, and these are the same treatments 8 weeks after the spray. So you can see we have a good control here as compared to the non-treated. And we are also conducting some of the herbicide studies with the different pre and post herbicides in corn. So water hemp, as I already discussed, this is the picture near the Rochester in the farmer field. This is the amount of infestation. So we are conducting a bunch of pre and post herbicide studies in the corn. Thank you.
Time for 1 or 2 questions.
Thank you very much. Certainly at the higher rates in the last pictures that you showed us, you're getting good control, but can you discuss what that means both economically and environmentally to go to those higher rates?
Economically, we'll have it will increase the cost, of course, and
for this particular chemical?
I don't think so. Yeah, because metribuzin is a very common herbicide and it's not very costly. Environmentally, there are many studies that have been people are conducting with different herbicides that how the soil microbiota is affecting, but we are not conducting the soil microbiota studies with that. Yes.
So you mentioned at the beginning of your presentation that there hasn't been much improvement on weed management for like 30 years. What are kind of like the big picture reasons for that?
Like I said that there's no new mode of action like herbicides. So it is because it is very costly. It has to spend like millions of dollars for the new mode of actions. So that is why the companies like Bayer and Corteva, they are not releasing new herbicide. They are just mixing different mode of actions like HPPD with the atrazine. They are releasing the new formulations instead of new chemical because it's very costly and it's very time consuming. It take like 7-8 years to release. Yeah.
Okay. So hello everyone, my name is Chenxi Feng and I'm currently a first-year PhD student in Dr. Ichiro's lab. And today I'm going to talk about my research project, which is about a apparent versus intrinsic temperature sensitivity of soil respiration. So why soil respiration? Because it's almost the largest terrestrial carbon emission, which emits about 60 to 80 petagrams carbon per year from the land into the atmosphere. and which makes up almost half of the total terrestrial carbon efflux. And in the meanwhile, it also affects the soil organic carbon storage capacity. And more importantly, the carbon emitted from soil respiration is growing rapidly through year, which indicates its higher and higher importance in global carbon cycling. So how soil respiration will respond to changing climates, for example, the changing temperatures or precipitations. Intrinsically, if we consider the chemical essence of soil respiration, it's basically a bunch of different organic compounds that are oxidized to produce carbon dioxide and other products. And this process is highly influenced by the temperature. And one explanation is the Arrhenius theory, which is shown in the left figure. So in this theory, the temperature will not only influence the molecular energy distribution, but also it will influence the activation energy, which is the EA in the figure, and hence it will influence the chemical reaction rate. And makes the reaction rate increase exponentially with temperature. But if we consider the enzyme catalysis in these reactions, the processes may be more complicated as shown in the upper left figure. So in these cases, there may be optimal temperatures for the reaction rate to reach the maximum. So other than these various intrinsic temperature sensitivities of soil respiration, there are also multiple environmental constraints such as the soil moisture and soil aggregation, which can modify the intrinsic temperature sensitivity of soil respiration. And moreover, if we consider the collective response from multiple processes in soil respiration, the observed temperature response, which is the apparent temperature response, may be much different from the intrinsic ones. So the question here is, should there be one or multiple functions for the intrinsic temperature sensitivity in different ecosystems? So several arguments for this question. There are several cases if the biochemical pathways governing the soil respiration and soil organic carbon compositions are all similar in different ecosystems, then there should be like similar intrinsic temperature function, but which is also assumed in most of the biogeochemical models. But in the real world, the case may be different because they may have different biochemical pathways and organic carbon compositions, which can lead to quite different intrinsic temperature functions. And to solve these questions, we've collected the soil respiration observations from several incubation experiments, and the results do show like different temperature response for the soil respiration. So in some cases, the soil respiration increases exponentially, as shown in the right figure, with temperature. But in other cases, the soil respiration may respond sigmoidally or unimodally to the temperature. So based on these results, we would like to calculate the intrinsic temperature response from the incubation data to see if there's universal function type or there are different kinds of intrinsic function types for different ecosystems. And the follow-up question is that if there's different intrinsic temperature functions, what's the reason cause this difference? And after we get this information from the incubation data sets, we can furthermore incorporate this temperature function into the biogeochemical models to further simulate the field measurements of soil respiration. And that's what we are going to do in the near future. Okay, thanks for your listening. Any questions, any suggestions? Yeah, please.
Interesting stuff. I was wondering what you're thinking about the timescales at which the incubation occurs versus some of the timescales that you're interested in. In other words, is short-term warming over the course of, say, hours or days, is that going to have different processes than, say, responses to longer-term warming?
Yeah, I think timescale is quite an important factor to influence the temperature sensitivity because they will sometimes influence the substrate availability, the carbon quality, such kind of things. And the incubation data set we have right now, our cooperators collected right now, focusing mainly on short-term incubation, not like for incubation for several years or decades. So basically, the incubation timescale is from several months to the longest maybe 1 year or 2 years, and not so long-term response, not so long-term timescale. But I believe there may be different temperature sensitivities in a longer timescale. Yeah, please.
This is in the nature of carbon between the different experimental units, or were they all similar?
Sorry, could you please?
CO2 evolution from your soil incubation, that's going to be based on the carbon compounds that you have in the soil, right?
Yeah.
So were they equivalent across your different treatments, or are you looking specifically at the behavior of different complexities of carbon within your system?
I see. So basically, I'm working on like the mixture of all kinds of different carbon compounds. So currently, we cannot like separate different carbon substrates and to see if they have different temperature functions. So basically, in the incubation data set, they have a soil sample which is sampled from a particular set, and it's kind of hard to separate like different kinds of carbon in the soil. So, but I think that's a good idea. Maybe in the near future we can work on that.
Okay, thank you. Right. Well, good afternoon, everyone. Before I start, on behalf of my peers, I want to this is an awesome turnout. Like, look at this. This is awesome. Yeah, it really means a lot for everyone showing up, supporting the first-year cohort. Right, community is so important right now, as Dr. Buckley alluded to. So this is awesome to see everyone showing support, right? So my name is Nazir Adam Sharifi. Feel free to call me Adam. I'm a first-year MS PhD student in the Sustainable Cropping Systems Lab under Dr. Matthew Ryan. And I'm excited to follow Chenxi's presentation and give an introduction to my own research on no-till corn production. So in the Sustainable Cropping Systems Lab, we do a lot of work in organic no-till rolled crimped systems. Essentially, what that means is we plant a cover crop in the fall, allowing it to grow in overwinter until it reaches a suitable growth stage for termination. Then we go in with a roller crimper, which is a steel drum with blunt blades mounted on front of a tractor to terminate that cover crop while simultaneously planting our cash crop with a no-till planter. And then after a few months, if all goes well, the goal is to have a healthy cash crop with the mulch serving as a protective layer to increase water infiltration and reduce runoff. The mulch also acts as a physical barrier to suppress weed growth without the use of tillage. So based on our research, we truly believe that no-till farming through the integration of rolled crimped cover crops has great potential to enhance field crop production, not only for organic farmers, but for the broader agricultural community. And currently, there's a wealth of studies that exist on no-till soybean planted into cover crops, with our lab contributing heavily to that literature. However, research on no-till corn planted into cover crops remains limited, with previous work facing systems and meeting the cropping system's demands. So, in the Sustainable Cropping Systems Lab, we developed a 256-plot surface response experiment to explore organic no-till performance under different cropping system designs. This approach is valuable for understanding how multiple variables such as nitrogen rates and seeding rates interact and influence response variables like weed biomass or corn yield under different rolled crimped cover crops. So in our study, we examined field corn planted into 2 rolled crimped cover crop mixtures, a hairy vetch oat and a hairy vetch cereal rye mixture. We also tested the impact of supplemental weed management through the presence and absence of inter-row mowing. So the inter-row mower features 5 individual mowing units that's designed to cut between the crop row and operating above the mulch surface. And additionally, we evaluated a range of corn seeding rates and side dress nitrogen rates. So there's a lot of things that one can do with such an awesome data set. And one avenue I'm exploring is a relatively new approach to analyze surface response experiments, and that's piecewise structural equation modeling, or PSEM. I'm particularly excited about PSEM because it provides a holistic view of the agroecosystem, making it ideal to understand ecosystem interactions. It's a tool widely used by pure ecologists. So what you see here is a simplified model of the interactions that we expect to occur in our organic no-till corn system. Let's take a quick look at how these factors are connected. So increasing corn density boosts yield, as does increasing side dress nitrogen. However, side dress nitrogen can also benefit weeds, which increases weed biomass. And then more weeds negatively impacts yields, while higher corn density suppresses those weeds through competition. And this is the general framework, but what happens when we apply this to a hairy vetch oat system with in-row mowing? What about without in-row mowing? How do these interactions begin to shift? You can take a quick look at some preliminary results. So let's start by looking at the hairy vetch oat system without in-row mowing. In this model, a solid line indicates a significant effect, while a dotted line represents a non-significant effect based on p-values. So in the hairy vetch oat system without in-row mowing, side dress nitrogen doesn't significantly affect weed biomass. But what I want to focus on more here is this finding, the significant negative impact of weed biomass on corn yield. This reveals a key system constraint. While hairy vetch, a legume cover crop providing high nitrogen to that corn, it lacks sufficient above ground biomass during the field season to effectively suppress weeds. But by adding the in-row mower for supplemental weed management, we achieve an exciting outcome where weed biomass no longer significantly affects yield. These preliminary findings validate the in-row mower as an effective tillage-free weed control tactic for field corn. And for simplicity, I previously omitted p-values and standardized coefficients, but they offer very valuable insights, right? These coefficients quantify the influence between system components. So here we can see that corn density and side dress nitrogen have relatively similar impact on yield, as shown by their comparable values. But how, again, might this change in a nitrogen-starved system where maybe hairy cereal rye is integrated into that cover crop mix, right? Will yield become a more strong driver of yield in this system? Like, these are the questions that we can begin to answer with PSEM. So moving forward, I'm excited to dive deeper into this data and carry out a streamlined version of the study to examine these interactions across different site locations. And thank you so much for your time, and these are my acknowledgements. More than happy to take any questions if we have time.
When you do the roller creeper and you have a mix, is it hard to kind of nail? Because I know like if you have just one species, you need to be at the right time. But if you have a mix, then it's kind of
Yeah, you're hitting me on all the spots that hurt. Yeah, timing is critical, right? And when you have a mixture, you're trying to aim for critical timing of termination. If you don't terminate it at the right time, it can come back up, or if you terminate it too late, it can reproduce seeds and infest your seed bank. So timing is critical. And yeah, it's kind of like in hairy vetch oat and a cereal rye mixture, we try to aim between 50 to 100% anthesis of that cereal rye, and then mid to late that flowering stage of that hairy vetch. So it's kind of finding the nice in between of where can we get the best kill of both of them. But excellent question, yeah. Enrique.
Notice any effect on crop emergence? The roller.
Wow, these are good questions. Yeah. Emergence is something that I'm actually really interested in. So I was an undergrad actually at Cornell University and research assistant in the Ryan lab freshman year, and something I see a lot around our experiences, emergence is always an issue, and I am really maybe technologically optimist here, but hoping that some good planting technology can cut through these thick cover crop mulches, because they provide so much, as I said, weed suppression and water infiltration and soil erosion, which is a huge problem, especially in the Northeast. So technological optimist maybe, but yeah, emergence is something to consider.
In one of your figures, I was just thinking where you see an improvement in corn yield or whatever crop yield at the end and potential decrease in weed biomass, you include the inter-row mower. Do those benefits outweigh what would be the additional costs of actually renting or owning an inter-row mower for a farmer?
Great question, right? If it's not profitable, why even use it? I really hope to conduct a profitability analysis. I feel like all experience now is like so important, right? If it's useful for the farmer, it has to be profitable, right? So that's something I'm hoping to look at in terms of like a profitability analysis, comparing the different treatment combinations.
Off the top of your head, like which combination of like cover crop, nitrogen, inter-row mowing, not inter-row mowing, like led to the highest yield?
Wow, good question. Yeah, so I can maybe. So it's hairy vetch oat treatment combined with inter-row mowing at a seeding rate of 45,000 seeds per acre and a nitrogen rate of 56 kilograms of nitrogen per hectare.
I'll do that.
Yeah, do that. Do that and you'll get hopefully perfect yields. Yeah, but maybe I should preface, like this is in the very early stages, right? No-till organic corn is not widely practiced, so that's why this experiment, this stuff is important to maybe push it forward and fill the gaps of knowledge that we have in this system. So, thank you.
This has been a production of Cornell University, on the web at cornell.edu.